class? How you guys doing? I'm going to pick up where we left off from last time. We were talking about DNA replication. Now the spring break's over. I gave you guys a week, so we're just going to kind of pick up from there. We're talking about DNA replication. We're going to transition from DNA replication into mutations. So just like the assignments that we've posted in Google Classroom. So hope everybody enjoyed this spring break. And here goes. Where we left off, we talked about DNA replication. We talked about mRNA. We talked about the messenger RNA that actually takes place here in the cell nucleus. We talked about the message that is actually written as a part of DNA replication. It is imperative that you guys remember. And when we talk about DNA replication, all right, remember what I told you, and Monica told you, I said Gotham City is where it's at. Here's a perfect example of a DNA base. We say Gotham City. Guanine, cytosine, all right, once again, they pair up. Those are your base pairs. Remember, held together by hydrogen bonds. Here's one example of base pairs. AT, all right, adenine, thymine, once again, held together by base pairs. Now, that's in DNA. That's when the message is actually being written. When it comes down to the muscle, the RNA, when the proteins are being synthesized, then all of a sudden, it changes up a little bit. And it can be kind of confusing, so let's make sure that you pay attention. The G and the C are going to stay the same. You never have to worry about that, okay? Guanine, cytosine, we're going to stay the same, okay? But in the DNA base, if there is adenine in the DNA base, that means that the RNA complement, we're going to introduce a new one, is going to be a U. It's called uracil, okay? So once again, that's only in the RNA complement. In the RNA complement, if you see in the DNA base, if you see adenine, you see an A, then it's going to be complemented with a U. However, if the DNA base has thymine, has a T, all right, you still use adenine. So it can get kind of confusing. Let me know if you guys have any questions on that one. But remember, when we talk about RNA complement, we're going to replace the T with the U for now. G and C stays the same. So, like we said before, we talked about the message being written, the tools being supplied by an RNA, by the tRNA, and transferred into the ribosome, which actually produces a number of proteins. Some of the proteins that we actually talked about, leucine, for example, I'm just giving an example. Leucine is important for skeletal muscles. It's important for exercising skeletal muscles. It's very important for cross-country athletes to understand that. Isoleucine is kind of like an extension, so to speak. Once again, it's another important amino acid that's not only important for skeletal muscle, but it's also important for muscle tear and rebuild and things like that. It's important for weightlifters as well. So a lot of this stuff actually applies to everyday life. There's even arginine, which, you know, with arginine, it's important for blood flow in the blood vessels and everything. And my guys might want to learn to appreciate that one a little bit, because if you do your research, you see how important it is. Wink, wink. All right, so now, as we go over here, we're going to transition into different types of mutations. And once again, a mutation, if nothing else, it's a simple change. Not simple, but it's a change in DNA. The thing is, we watch a lot of movies, you know, we see a lot of mutants in a lot of movies and everything. And, you know, there's a little bit of truth to it, but not quite to the point of that we see in entertainment. And we're going to kind of explain some of that today. So, like we said before, some of you have seen this particular, in one of the assignments I gave you, some of you have seen this particular amino acid, alanine. Our DNA base was cytosine, guanine, and adenine. So our RNA complement was guanine, cytosine, uracil, GCU. That's going to give us alanine, just like in your test. All right, just like in the assessment that I gave you. For anybody else who's watching, they also have this here to kind of help them in everything. If you need me to send that to you, for anybody who's watching, need me to send this to you, it has a number of different amino acids and proteins that are a result of such a process. Okay, so once again, we got 1, 2, 3. Remember, these are called codons. Okay, these are called codons. And what we got here, like we said, we got alanine. Sometimes these codons change, and there are different types of changes. I'm going to go over the first one. Okay, sometimes these codons change because of surrounding conditions, adaptation to surrounding conditions. That's going to lead into our next unit when we talk about evolution. But there are some surrounding conditions that can change that particular one. And then sometimes there are extensive chemicals, okay? So that's why I always say that we have to be careful of what we actually take into our body, especially when it's pre-cooked food and things like that. Hold on 1 second, please back up, thank you. I'm sorry, we got some antsy children over here. So once again, like I said before, chemical intake, all right? So I always say be leery of pre-cooked foods, things like that. Not all of them are bad, but we want to be cognizant of that as well because it takes certain chemicals to actually preserve them. So chemical intake as well as intensive radiation around us, and that kind of plays into you not being able to stand out in the sun for too long, at least without sunblock. Why? Because that's intense radiation, and after a while you can develop, certain changes within the body. All right, so one particular mutation I'm going to talk about today is a substitution. Okay, all right, a substitution. One example of a substitution: sickle cell anemia. Now, a lot of people refer to it as sickle cell disorder, sickle cell anemia, things like that. Not so much. A lot of people don't quite know the history on it. A lot of you do, some of you don't. All right, the thing about sickle cell anemia, for those of you who don't know, it's an oblong sickle-shaped formation of the red blood cells, which prevents blood flow, which hesitates blood flow, resulting in pain and discomfort, amongst other things. However, most people who actually go through this particular condition, as you guys know, are from tropical environments. Okay, the thing is, in those tropical environments, malaria is also often rampant in certain areas. Okay, well, those with the sickle cell trait and those with the sickle cell condition, a lot of times are, I'm not going to say immune, but they have greater immunity to this particular malaria, and it's an adaptation. It's believed to be an adaptation to the malaria in the original surrounding area. So what we have here, and when that actually happens, is we have a sequence. We got a DNA base. Our RNA base here is G, A, and C. That's glutamic acid. All right. Well, when that changes, all right, and that changes like here, it changes. All of a sudden, it switches up and it switches to valine. That means that now the different. And let me know. I've also sent this in an assignment as well. All right. That means that the sequence now has changed up. It results in that particular sickle cell condition, which once again is originally believed to originally designed in order to protect from conditions such as malaria. Another example of these different types of mutations and change that we talk about that apply to everyday life, we got what is considered non-disjunction. All right, some people know where I'm going with this. All right, once again, we got like guanine, cytosine, we have adenine, uracil right here. But the thing is, we have two G's. Sometimes two guanines. What happens sometimes in this particular situation, and it's more common with the age factor, what happens sometimes in these particular situations is that when meiosis takes place on that 21st chromosome, all right, there's a failure of these chromosomes to split. And what happens a lot of times is that you get an extra unit here that has no complement. When this often happens, is referred to as, we commonly know in layman's terms quite often, as Down syndrome. And a lot of times this particular condition can be picked up on within the 10th to 13th week of pregnancy. So this is how a lot of these particular concepts actually apply in real life. So once again, we have a substitution, which we talked about here. Sickle cell is an example of a substitution, but then non-disjunction is another example. And then there are other changes, other mutations that we also refer to as deletions as well. Deletions are often responsible for conditions such as muscular dystrophy, also responsible for conditions such as cystic fibrosis, male infertility. All right. So once again, a deletion. In other words, when a DNA base is actually removed, a lot of times, once again, these particular conditions, they are the result, more so the likelihood of them are increased with age within the individual. So once again, that kind of sums it up for the most part. If you guys have any questions, you guys can always email. I definitely have this codon chart that tells you all the different types of amino acids and the different foods they can actually be found in. So, I always say meat's not always the enemy, but, some people beg to differ. So anyway, if you have any questions, if you have any concerns, please shoot me an email. I'll do the best I can to get back to you as soon as possible. Please keep in mind that during this pandemic, not only am I the teacher, but I'm also like the groundskeeper and the cafeteria manager as well, so it takes away a lot of my time. All right, but I get to you as fast as I can. I hope all is well. Let me know if you have any questions. Thank you.